Our names are Madeline Fike and Douglas Lee, and we are presenting conducted research for the influence of genetic background on neurological responses to viral infection. Our faculty advisor is Dr. Brinkmeyer Langford, and our team leaders are Cody Lawley and Araceli Perez Gomez.
So neurological diseases such as Parkinson's disease and multiple sclerosis may be the result of an earlier viral infection. We utilize Theiler's murine encephalomyelitis virus, or TMEV for short, to understand how a single virus can contribute to different types and severities of neurological disease outcomes. In mice specifically, TMEV infection elicits diverse neurological conditions in mouse strains with diverse genetic backgrounds, suggesting that genetic background of the individual influences the neurological disease characteristics observed.
To further our understanding of how genetic background impacts neurological damage in response to viral infection, we aim to understand the differences in neurological disease symptoms we see in genetically distinct mice.
In the present study, we employ a specialized mouse model known as the collaborative cross, or CC. It's important to note that the CC models are utilized because they are more reminiscent of human genetic diversity. CC mouse strains were developed using 8 founder strains of mice that were bred and crossed for generations in order to achieve maximum genetic recombination. Each CC strain is a genetic mosaic of the 8 founding strains as observed in Figure 1. Historical populations of mice used in studies are inbred and therefore do not possess heterogeneity. This results in stereotypical behaviors and phenotypes, which is beneficial for studies involving experimental replicability, but not to study genetic variation. As modeled in Figure 2, the CC strains have more variation in gene expression, which induces variation in observed phenotypes.
The experiments that we are involved with the most are quantitative and qualitative phenotyping over the acute and chronic phases of infection. These are performed in order to observe neurological changes in the mice over time as a result of viral infection. Figure 3 lays out the experimental timeline from TMEV infection to tissue collection. Mice of varying strains are infected at 0 DPI or days post-infection. The acute phase measures neurological deficits that are sudden in onset. The chronic phase of infection is significant to observe because humans often develop neurological diseases such as multiple sclerosis later in life, not right after they get the virus. Qualitative phenotyping methods were performed twice daily until 14 days post-infection, and following that, they were performed once weekly until 90 days post-infection. We looked for things such as limb weakness and paralysis, hunch, seizures, squinted eyes, and reflex. Reflex tests for the ability and speed for the mouse to right themselves onto 4 limbs when turned onto their back. A slower reflex and severe weakness or paralysis, as well as a combination of other phenotypic deficiencies, are good indicators of neurological symptoms associated with multiple sclerosis, Parkinson's, or other neurological diseases.
Quantitative phenotyping was performed at 3 time points indicated by asterisks in Figure 3 and includes motor evaluations performed using digigate imaging and rotarod analysis in Figures 4 and 5 respectively. Neurological symptoms of MS and other neurological diseases are often visible in the way that the affected individual walks. Digigate imaging uses a camera that records walking patterns in the mice such as stride length, braking time, propulsion time, and swing time while the animal walks on a treadmill. Rotarod is used to measure parameters such as endurance. It is often indicative of the mouse's motor coordination, balance, and strength. Typically, severely affected strains struggle to stay on the rotarod due to hind limb weakness and paralysis, hunch, and loss of motor coordination. Once the mouse reached 90 days post-infection, tissue samples were obtained from the brain, spinal cord, heart, and spleen for histological analysis and RNA sequencing.
Figure 6 displays the phenotypic profiles of several different mouse strains. As clearly seen, different strains show very different phenotypic profiles. The frequency of phenotypes also vary from strain to strain. Strain K displayed a high frequency of limb weakness, while strain A experienced less limb weakness but showed more reflex abnormalities, hunch, and limb paralysis than strain K. Figure 6 displays strains that develop significant neurological conditions, but there were also infected strains that showed relatively normal motor functions throughout the experimental timeline. To further understand the development of neurological conditions within certain strains, brains were cross-sectioned coronally to determine if there was an association between the lesions on the brain and the outward phenotypes observed. In Figure 7, 3 coronal cross-sections of 2 stains are illustrated, with the top row indicating one strain and the bottom row showing another. The different colors indicate lesions of different animals within the same strain. A single color on a specific cross-section of the brain indicates similar lesion burdens within the strain and low variation. The top strain shows more thalamic lesions than the bottom strain, but generally the hippocampus and thalamus exhibit the most virally induced lesions.
We found that genetic diversity of CC mouse strains following infection with TMEV results in a wide variety of central nervous system lesion locations and phenotypic differences. In the future, we will further evaluate how the location and severity of central nervous system lesions look different across differing mouse strains following TMEV infection to better understand the development of virally induced neurological diseases, as well as how phenotypic outcomes and lesions differ in male and female mice. We are interested in mapping out the locations of lesions and performing further behavioral analysis to determine if there is a correlation between specific lesions and specific phenotypic outcomes. We will also look at the viral load 90 days post-infection and see if there's a difference between the central nervous system and systemic viral load. Ultimately, we would also like to determine whether or not there are quantitative trait loci present so that we can determine if there are any gene locations associated with the genotypic traits.
With that being said, this concludes our presentation over the influence of genetic background on neurological responses to viral infection. We would like to thank the Aggie Research Program for allowing us to be involved in undergraduate research, Launch Undergraduate Research for hosting the Fall Undergraduate Research Expo to present our experiences, and Dr. Brinkmeyer Langford and her funding sources for giving us the opportunity to become involved in her research at Texas A&M. Thank you.